Welcome to this explainer. Today, we're diving deep into a microscopic structure that acts as both a life-saving vaccine component and a deadly toxin. It's a true biological paradox, hiding right on the surface of some of the most common microbes on Earth. So, I want to start things off with a pretty intriguing question. What makes a dead bacterium just as dangerous as a living one? Well, to answer that, we have to look closely at a remarkable molecule called lipopolysaccharide, or LPS for short. It's the key to this whole fascinating and honestly pretty dangerous biological reality.
Here is our agenda for today. We'll start by meeting the bacterial fortress, then look at the O antigen shield, the connecting core, lipid A, which is the deadly anchor, and finally, what happens when bacteria break down.
All right, Section 1. Meet the bacterial fortress, an overview of gram-negative bacteria. As you can see, LPS is an amphipathic molecule that's completely embedded right there in the outer membrane of gram-negative bacteria. It's doing some heavy lifting, providing vital structural support to maintain the integrity of the cell wall. Basically, it ensures the bacterium actually survives under severe environmental stress. Moving on to Section 2, the O antigen shield, the outermost protective layer. Now, sitting at the very top of the structure is the O antigen, which is fully exposed to the extracellular environment. This thing is a repetitive glycan polymer packing in over 100 oligosaccharide units. And what's wild is that it's a highly heterogeneous shield. It varies wildly between and even within different bacterial species. Because of this, it acts as the primary site of immunologic specificity. What makes this so fascinating is how human medicine has actually hijacked this exact shield. You see, because the oocyte chain is so exposed and unique to specific microbial serotypes, it is an absolutely vital tool for human vaccines. We literally take the bacteria's own identification shield and use it to program our T memory cells, delivering long-lasting secondary immunity. How cool is that? But how exactly do these bacteria build such a complex shield? Well, they manufacture it right there on the membrane in 4 rapid enzyme-driven steps. First up, you have the initial assembly of lipid intermediates. Second, the polymerization of those units. Third, the enzymatic transfer of those fresh O polymers directly onto the LPS core. And finally, the regeneration of the carrier lipid so the whole cycle can fire right back up again. Next up, Section 3, the connecting core, bridging the gap. Sitting adjacent to the deepest part of the molecule, we find the core polysaccharide. It's constructed by membrane-associated enzymes that carefully catalyze the addition of monosaccharides. Basically, this core builds a very sturdy molecular bridge right down toward the base of the cell wall. Now, a totally crucial component inside this core is a specialized acid known as KDO. You can really just think of KDO as the definitive molecular bridge. It forms a ketoacidic linkage directly to the inner lipid A substructure, so it's essentially acting as the heavy-duty lock that completely secures the exposed outer core to the innermost anchor of the bacterium. That brings us to Section 4, Lipid A, the deadly anchor, the hidden, highly conserved base. Okay, meet Lipid A. This is the substructure deeply embedded within the bacterial cell wall. And yeah, while its 3D structure might actually look a bit like common household soap, please do not let that mundane comparison fool you. Its biological function is intensely formidable, and it's absolutely central to the danger these bacteria pose. Lipid A is actually the most structurally conserved part of the entire LPS molecule. This deeply embedded anchor features these long 16-carbon fatty acid side chains, and it is incredibly robust. I mean, purified lipid A can literally withstand extremes in both heat and pressure, all while keeping its biological activity completely intact. Finally, Section 5. When bacteria break down, the biological irony. So we know what the fortress looks like now, but what actually happens when our body's cellular network successfully destroys these invading gram-negative bacteria? You'd logically think, hey, destroying a bacterium is a pure victory, right? Well, here is where our structural tour turns into a bit of a medical thriller. As those gram-negative bacteria are attacked by our cellular network, their cell walls are lysed or broken apart. This rips the LPS molecule apart, which releases that extremely durable lipid A anchor directly into the host organism. This free-floating lipid A then immediately binds to effector cells like macrophages, and that is what triggers a massive pathogenic cascade. The scary reality here is that this single molecular anchor is the true pyrogenic endotoxin. At lethal doses, its release into the body results in rapid fever, profound hypotension, and even fatal septic shock. So by successfully killing the bacteria, the body inadvertently unleashes the very toxin that does the absolute most damage. Talk about a catch-22. And because lipid A can withstand that extreme heat and pressure we talked about, it remains highly dangerous long after the bacteria are dead. The pharmaceutical industry simply cannot afford to take chances here. They rely on strictly published, highly sensitive techniques to detect this exact endotoxin in liquids and on medical devices. Think about it. A perfectly sterile liquid could still contain deadly lipid A if it isn't properly screened. And so we're left looking at this microscopic fortress that literally both cures and kills. The LPS molecule really is a masterclass in biological complexity. The big question moving forward is, how do we continue to utilize the highly specific, life-saving shield of the O antigen to create vaccines, while simultaneously and perfectly disarming that deadly, durable anchor of lipid A hiding just beneath it? It's a massive challenge, and one that continues to drive modern immunology today. Thank you for joining me on this explainer, and I hope you never look at a microscopic cell wall the exact same way again.